Hi everyone, I'm Hasiba Karimi, a fifth year medical student at Bezmane Vakıf University, Istanbul, Turkey. Thank you for joining my session. My talk is on studying the dynamics of Ebola virus proteome sequence diversity. Ebola virus disease, which is common among humans and other non-human primates, is a deadly disease that causes a highly fatal hemorrhagic fever. It's hemorrhagic because of the clotting problem it causes, leading to internal bleeding. Herein, we describe a proteome sequence diversity analysis of Zaire Ebola virus, or ZEBOV in short, an agent responsible for Ebola virus disease. ZEBOV is the most pathogenic among the identified species of Ebola virus. The methodology employed for our project is as follows. A total of 23,543 ZEBOV sequences were downloaded from the NCBI virus database for the human host, which was deduplicated by CD-HIT. BLAST-B separated into 8 encoded proteins, and each was then aligned separately by use of MAF-DON muscle. Shannon's entropy and motif diversity analysis were performed by use of DEMA tool for each of the aligned overlapping monomer positions to measure the proteome diversity. As part of the motif analysis, sequences at a given monomer position were grouped into 4 motifs, namely index, major variants, minor variants, and unique variants, based on their incidence. Let's briefly define the 4 motifs. The sequence with the highest incidence, that is the most common one, is indicated as the index monomer, while the others are referred to as its variants. Define the sequences that are different from the index monomer by at least one amino acid. All the variants at a position, or total variants in short, can be further subclassified into 3 variant motifs: major, minor, and unique. The major variant is the second highest incidence monomer at the position, and hence the most common variant. Minor variants are sequences that occur more than once in aligned viruses, but with an incidence of less than or sometimes equal to the major variant. Unique variants are singletons, each observed only once in the alignment. Now let's dive into the results. We started with 23K sequences for ZEBOV. However, after the duplication, it dropped to 2,706 unique sequences. This is a huge drop of 88.5%, indicating a high redundancy in the database sequences. As you can see here, Figure 1 provides the ZEBOV entropy plot and incidence of total variance. The entropy is illustrated by black color on the left y-axis, and the incidence of total variance is shown by pink color on the right y-axis for each of the aligned monomer positions. Higher entropy values indicate higher diversity, and the lowest entropy value of 0 indicates complete conservation. Total variance is defined as simply 100 minus the incidence or frequency of the most common sequence, which we refer to as the index. For example, let's say if a position has a predominant sequence or index with an incidence value of 90%, then the total variance incidence at that position would be 10%. The ZEBOV proteome was highly conserved with a mean entropy value of 0.2. The envelope glycoprotein, shown here as GP, was the most diverse one with an average entropy of 0.38, while VP30 was the most conserved with an average entropy of 0.12. The highest entropy of 1.84 was observed in GP, with the peak incidence of 54.69% of total variance in small secreted glycoprotein. For comparison, the envelope protein of the extremely diverse HIV-1 clade B of entropy 9.2 and total variant incidence of 98%, shown above here as lines, is used as reference, as it is the highest monomer entropy value that we have observed from our various analyses of viruses. This signifies the high conservation of ZEBOV proteome relative to the reference. Now, Figure 2 illustrates the relationship between the entropy and total variance of ZEBOV monomer positions, with each dot representing monomer position. A positive correlation is observed, however, with a wider spread in total variance with increasing entropy or diversity, particularly from entropy value of 1 onwards. So an entropy value can have a wide range of total variance. Given this, we then went on to investigate the diversity motifs that comprise the total variance, namely major, minor, and unique variants mentioned earlier. Figure 3 provides distribution for each of diversity motifs. Each variant motif, major, minor, and unique, have their own distinct pattern. In these plots, the x-axis shows the total variance, and the y-axis indicates the incidence. The incidence of the predominant sequence that is shown in black here in the index range from 100, which means completely conserved, to as low as 45.30%. Major variance, which is shown in green here, showed a restrictive pyramidal pattern with a peak incidence of 48.63%. Notably, minor variants, which are shown in blue here, were limited to the highest incidence of only 23%. While unique variants, which is shown in violet here, were present at most of the positions with non-zero total variance, with the highest incidence of 4. Nona types, as you can see here, evaluate the multiple distinct variants, with most of the non-conserved positions exhibiting a variant, and the highest incidence of being 5.11. Violin plot in figure 3e depicts the frequency distribution of the incidences for all motifs. It should be noted that the complete conservation was observed for 18% of the proteome positions for EBOV proteome. In conclusion, the results herein offer new insights that help better understand the sequence diversification of the virus with implications to vaccine and drug design. Thank you for your attention.